Welcome to the third part of cell membrane and transport processes. We will start with talking about transport via compartments. We talked about simple diffusion, diffusion with facilitate, which is carriers. These are the examples with small molecules easy to cross the membrane themselves, which is simple diffusion, and molecules slightly bigger, so they can't cross the membrane easily. They need a carrier, which are specialized protein. So this is like you can't swim across the river, so you need a boat. And if it's an ion channel, then it's like you build a bridge over the river. And if it's not following the concentration gradient, if it's against the concentration gradient, then you need to consume energy. So you need to consume ATP. So that is how the primary and secondary type of active transport come in. What if the molecules being transported are super big, like the macromolecules? In that case, we need to get the trench of the membrane involved, so which is compartment transport. So in this membrane, use the membrane compartments, we need to divide them into roughly two parts. First part is endocytosis, which is transport the molecules into the cell. And the other part is exocytosis, which involves transport the material out of the cell. So let's look at the endocytosis first. So endocytosis, there are three types. The first one is phagocytosis. So if the molecule is a solid particle, and when this comes into the cell membrane, the membrane is involved into a vesicle, and this vesicle come into the cell and become a phagosome, and this phagosome is binding the materials and then leave the lysosome. So this part that involving the compartments and transport a particle into the cell is called phagocytosis. The next one is pinocytosis. That involves the dissolved molecules in a similar way, involving the compartment that is changing of the membrane and become endosome. So that is a pinocytosis. The third type is called receptor-mediated endocytosis. So in this one, you have molecules that are being transported will be combined with receptor proteins embedded on the cell membrane, and those molecules being recognized by the receptors and then evolving into this form clustering, and this clustering going into the cell, and that would transport the molecules into the cell. So this is called receptor-mediated endocytosis. This is very specific. So the receptor-mediated endocytosis is a very important type of membrane transport. The main mechanism is the macromolecules will be recognized by body cells by interacting with a specific receptor that is embedded on the cell membrane. So the receptors, they binding with the substance and then internalized with the invagination of the plasma membrane. So that is called clustering-coated pin, which would form the endocytotic vesicles and then form an intracellular compartment. So that is similar to the phagocytosis and the pinocytosis. The difference is it is specifically recognized by the receptors. And this process is common to all eukaryotic cells except the mature red blood cells. So in the next one slide, you can see that there are a lot of materials being transported into the cell by this mechanism. So these are the example growth factors, for example, and cytokines and some hormones, specifically the peptide type of hormones, including insulin and luteinizing hormone, follicle stimulating hormone, growth hormone, and prolactin releasing factors. So these are the examples of hormones that is being transported into the membrane by receptor-mediated endocytosis. And also glucoproteins, immunoglobulins, and transcobalamin. These are the vitamin B12 transporter. Among them, cholesterol is another one. So cholesterol is being wrapped into LDH and HDL, LDL and HDL. So these are the high density lipoprotein and lower density lipoproteins. So these lipoproteins are being recognized by the receptors on the cell membrane, and then by involving into the cell, cholesterol is being transported into the cell. So that's how, and specifically the LDL is specified by depositing the cholesterol onto the blood vessel wall. So that is why it's easy to get deposited onto the blood vessel and form the atherosclerosis. But the high density lipoprotein deposit the cholesterol into the liver, so it's less risky for the body. The next type is exocytosis. So the exocytosis, starting with the substance being wrapped into a secretory vesicle, and this vesicle is being fused with the plasma membrane and with an opening here. So the membrane of the vesicle starts to become part of the cell membrane, and by doing this, the content of the vesicle is being released into the extracellular space. So this is called exocytosis. It's the opposite process as the endocytosis. But no matter whether it's endocytosis or exocytosis, they have common feature. First of all, they involve the changing of the membrane itself. That's why we call it transport by compartment. Secondly, they involve the use of ATP, so they are energy consumed, no matter which direction they go. So an example here is the release of acetylcholine at the neuromuscular junction. So the acetylcholine is wrapped in the vesicle, being fused with the membrane at the neural axon terminal. Then the acetylcholine will be released into the cleft to be binding with the ion channels on the muscle membrane. So if we involve both part of the exocytosis and endocytosis, then this is a process of transcytosis. So this starting from the lumen here, the molecules being coming into the cell by endocytosis and cross the epithelial cell, go to the other side of the membrane, which is connected with interstitial fluid. So here it involves exocytosis. So by doing the combination of endocytosis and exocytosis, the material is being transported from the lumen across the epithelial cell and being transported into the interstitial fluid. So the combination of this is called transcytosis. The next one we would like to talk about is how the cell membrane transports water. We will start with the concept of osmosis. So on the left side, you have a compartment which is separated by a membrane. This membrane is half permeable. That means it's permeable towards some molecules, but not permeable to other molecules. So in this particular case, the left side is glucose dissolved in water. The concentration of glucose is 100 millimolar. On the right side is glucose concentration with 200 millimolar, also dissolved in water. So the membrane in between them is only permeable to water, but not to glucose molecules. So this is time zero. What would happen when it's equilibrated on the right side? Both sides, the concentration is 150 millimolar. However, instead of glucose being moved from the higher concentration, which is the right, towards the left, what happened is it's the water molecules that moved from the left to the right. Therefore, the final concentration of glucose is equal on the left side and on the right side, but the volume of the solution is not equal. So the right side has more water. That is because the water has been moved from the left side towards the right side. So what has happened there? Because the membrane is not permeable to glucose, therefore the glucose molecules cannot cross the membrane. Only the water molecules can cross the membrane. So what do you think? What's the reason that it is coming from time 0 to time 1? So water moves from higher concentration to lower concentration. So where does the water have a higher concentration? When the glucose have lower concentration, you have more water relative to where you have higher glucose concentration. So in other words, starting with time 0, the left side glucose 100 millimolar has a higher water concentration than the right side with glucose of 200 millimolar. So that's why the water moves from left side to the right side, and that's how it comes when time equals 1, which is when it's equilibrated, it's balanced. So the right side has more water volume. So this is the base of osmosis. So the osmosis, that by definition is the net diffusion of water across a membrane, and the direction of the movement is from the higher concentration of water towards the lower concentration of water. The channel for the water to move is called aquaporins. They are very, very small channels, so the diameter of water molecule is about 0.3 nanometer. So this group of membrane protein called aquaporin, they form the water channels. They are everywhere on the cell membrane, so the water can cross them easily. Remember, water is polarized. That means they are charged, so they're not easy to come across the membrane easily. So they need this specialized channel to cross the membrane. And the movement is following the concentration. That is why it's not energy needed. So the way to determine the water concentration is the opposite as the solute that is dissolving in the water. So the higher the solute concentration is, the lower the water concentration is. So let's look at this example of water transport over the epithelium cell. So these ones are the epithelium cell. You can see that the arrows pointing from the epithelial cell to the interstitial fluid. So here, if we look at this slide, is when the solute is pumped into the interstitial fluid, the osmotic pressure of the interstitial fluid would increase because you have more solute there and the water concentration is decreased. Therefore, osmotically, the water molecules will be pulled across the cell into the interstitial fluid. And that's how the water is transported from lumen to the interstitial fluid. Let's look at an example in the case of cholera, where the virus would be infecting the membrane transport, and where intracellular has elevated circular AMP, where the chloride channel would be open and chloride would come from the epithelial cell into the lumen, and sodium ion would go with the chloride ion because of the electrical gradient, and water would be pulled into the lumen, and that's how diarrhea is happening, have water loss. So this is a mechanism of cholera that is very dangerous to have lost the electrical sites. So that's why this virus is dangerous. We also would like to talk about the cell volume regulation. We know that the membrane transport is very important to regulate the cell volume, and cell volume needs to be well controlled. If the cell volume is not balanced, then the cells cannot function properly. So with plants and prokaryotic cells, you have a cell wall which is rigid, but animals, they must maintain the osmotic balance, otherwise their membrane is fluid, so it's a fluid mosaic. It's easy to move around. It's not as rigid as a cell wall. That's why cell volume regulation is very important. So when you have the cell volume regulation, the crucial factors is you should have something intracellular which is not easy to cross the membrane. In this case is the proteins. So the proteins are not easy to go out of the membrane. And on the other side, you should have impermeable extracellular solute. For example, the sodium chloride. They are not easily cross the membrane, so you have a balance. The membrane is semi-permeable, so they are not easy for the proteins to go out of the cell and not easy for the sodium chloride to come into the cell. In this case, the cell volume has a base to regulate. And another thing that we measure how the cell volume is being regulated is called tonicity. Tonicity is a concept that for a solution affects the volume of the cell. So the tonicity is a combination of osmolarity and permeability. So osmolarity is the concentration of how much solid is being dissolved in the water, and the permeability is whether this molecule is easy to cross the membrane, and how the membrane membrane transport is being controlled. So if it's isotonic, that means intracellular and extracellular has a balance, so the cell volume is not changing. But if it's the extracellular concentration is hypotonic, that means you have too little molecules dissolved in the extracellular medium, then this would cause the cell to swell. That means to enlarge the volume, because the water would move from extracellular space towards intracellular space, from the higher water concentration to the lower water concentration. That's why the cell volume is going to increase. And if the cell is suspended in the hypertonic solution, which means extracellular has too much solute there, then the water would move from intracellular to the extracellular space. Therefore, it would cause the cell to shrink. So if you have cell culture medium, you should make sure that it's isotonic. So we have finished the membrane transport 3 parts, and we started with the description of structure of plasma membrane and how important the membrane transport is. So we talked about the structure, what's the backbone of the plasma membrane and what's been embedded, like the proteins, and how they are contributing to the membrane transport. And we talked about the different types of membrane transport. And we started with simple diffusion and how to quantify this. We will not examine you on how to quantify this to calculate, but you should be able to recognize which factors would make the diffusion easier and which factors would make the diffusion slower. So you need to understand it is in proportion to the concentration gradient, for example, and it is inversely proportional to the thickness of the membrane, and temperature. The higher the temperature is, the quicker the diffusion would be. So these are the things that you need to understand. And also important is the area of the membrane, how it's been contributing to the membrane transport. And we also talked about facilitated diffusion and primary and secondary active transport. We talked about endocytosis, exocytosis, and water transport and osmosis. So that's everything. Thank you for listening.